I'm going to talk about some of the things that we thought about and suggested, and some of the open questions, of which there are many, about how human genome editing ought to be thought about and regulated. And I think maybe best if this is a discussion, so I'm not averse to people raising their hands in the middle and asking a question there. That might be a good way to do that, so feel free to do that. So, as Dana said, I co-chaired this committee with Alta Charo, who's a lawyer and ethicist, who's already been mentioned by Hank, and perhaps unwisely, because it took more than a year. It started basically with the International Summit, which we were not responsible for, but we attended, and it was basically our launching pad. That was back late in 2015, and then we released the report on Valentine's Day this year. Same day that Flynn resigned or was fired, so that kind of gave us some covering smoke. So I want to talk about some of the issues. Now, I'm not going to talk essentially, well, a little bit about the science, but mostly about issues of ethics and regulation, and asking the questions, what can we do now? You've heard a lot about that, so I won't dwell on that. What could be feasible, and I will talk a little bit about that. And we've heard a number of things, and you've heard more than I did, about what could be feasible in the near or far future. What of those things should be done and what should not? I think that sort of encompasses the debates. So the specific charge that we had from the National Academies, this was both sciences and medicine, was to evaluate the state of the science, the potential for clinical applications. This was focused only on human. But we broadened it a little bit to the basic science because that's important to preserve that while not letting the worries about human research, clinical research, impinge upon, for instance, work on embryos in vitro. The whole question of ethical, legal, and regulatory standards and the societal implications of this. And because this was meant to be part of an initiative, well, it was part of an initiative to try and establish some international consensus about these things, to work out some overarching principles that could be applied to regulations in different cultures, because different cultures think about these things differently. There is little to no prospect that there's going to be an international set of regulations that everybody will follow. In a decade ago, I was involved in a similar thing about stem cells. And at that time, there were efforts in the UN to ban cloning. They came to nothing, human reproductive cloning, because nobody could really agree how to do that. And I think the same thing is going to be true here. Cultures can be very different, and in terms of what people think is important to worry about and important to do. So the idea was to get a set of overarching principles that everybody should apply, but they're going to apply them differentially. So that was what we were asked to do. And because of that, it was a very big committee with a lot of international representation. I think it was in certainly 4 continents. I forget how many countries, but it was a big committee with all sorts of different expertise on it, including ethicists and lawyers and genetic counselors, patient advocates. We were asked to produce a report, which we did. And so I want to go through a few of the topics and touch upon things that we've heard, some of them earlier today, which I won't repeat, but I'll just mention. So you don't need this. It was in my slide deck, and I decided not to take it out because it wanted to remind me that some of the changes that this has wrought, you heard about this morning, about gene therapy. These changes, which I know you've heard about turning things on and off and using things other than actually cutting the DNA, and the prospects for RNA editing and regulation, multiplexing and things like that, I think they're important because one of the questions that would be very useful in terms of thinking about regulation is whether one can reverse any of these changes. And embedded in that set of things are some of the possibilities, and that will come up a couple of times. So, as I said, we wanted to insulate basic laboratory research from the complications that come when you apply things clinically. Because there's a lot of very valuable stuff that can be done on editing, you all know that, but particularly on editing embryos and early germline cells. We all think about things in terms of the mouse, because that's been the experimental system, but early mouse embryogenesis is significantly different from early human embryogenesis. And there's a lot of stuff that we don't understand about human embryogenesis and early development because the mouse doesn't actually apply. And we know now that the regulatory systems are significantly different. I don't know how many of you are developmental biologists. That's what I am originally. Mouse development is significantly distorted from many other mammals, including humans and primates. That leads to a different set of regulatory circuits controlling the different early developmental events, like how you make a placenta, which parts of the embryo are pluripotent and which parts are not, and how that's regulated. So there's a need to understand how human development works to work on human embryos. And there's a lot of progress in that field. In parallel with the progress on CRISPR-Cas, partly using CRISPR-Cas as a tool, but a lot of developmental biology in the last 2-3 years, even 5 years certainly, that's making a huge difference. And I'll come back to that when we talk about germline editing. So this needs to be done to understand things like fertility, to understand implantation, to understand abortions and miscarriages and all those things to do with artificial reproductive technologies that would be much improved if we knew more about human embryos. So it's important for that to continue, where we're not talking about any implantation, no pregnancy. And in fact, there's no reason why that shouldn't, because the current regulatory systems cover that pretty well. It's not that they're free of controversy, they're not, because many people disapprove of doing work on embryos in vitro. However, that's been thought through in terms of stem cells and iPS cells and alternative reproductive technologies. And the regulatory systems are working pretty well. And CRISPR-Cas and TALENs and zinc fingers don't really alter that at all. They're just, the tools are better, but they're operating in the same context. So the regulatory system is fine. I highlighted this about the 14 days in vitro. I'm not going to talk about it very much, but that's now come up as a debate, which we're going to have to deal with. And it impinges on this because that rule's been in place for a long time that says you can't culture a human embryo in vitro for more than 2 weeks, or when the primitive streak, which is the formation of the mesoderm, when that happens, whichever comes first. And everybody's agreed on that. It's now becoming feasible to think about, and actually almost to do, but nobody's done it yet because you're not allowed to culture human embryos in vitro for longer than that, which would allow one to explore other aspects of those reproductive questions that I talked about. So that's going to be a big debate, and my guess is there'll be another report on that. It doesn't impinge yet, but it will very soon. So that's why I highlighted it in red. So this is a list of the things which I won't go through. You can read them, but I summarize some of them, for which work on human embryos and iPS cells and germline cells in vitro is very important and very actively being pursued. But it's well regulated now. So it's not particularly controversial. The germline editing has been discussed, and although not everybody agrees, it's in a stable situation. The second category, and we separated that germline editing in vitro from anything about heritable germline editing. Well, we wanted to make those things widely separated so they don't cross-contaminate. See if that works. So far, it has, but we'll see. You all heard about this, so I'm hardly going to go through this slide. You heard about it this morning. Obviously, this is not new from gene therapy. It's just better, faster, and easier. So the regulations that have been in place for somatic gene therapy, where you're editing tissues in the body, or ex vivo, editing them and putting them back, but not touching the germline, or making sure you haven't touched the germline. That was also covered this morning. Those have been well regulated. It's been worked out. It's been going on for 15, 20 years, and there's a good set of regulations. This doesn't really change anything. You heard from Don about the two different ways of doing it. It's still early days. There are not many of these things that are through to the point of application. There are some. There are quite a lot in clinical trials, but it's coming, without a doubt. But again, it's not particularly controversial. There's a lot of technical stuff to be worked out, and you heard quite a bit of that in a very nice talk this morning. So I was going to stress this more, but Don already went through all the difficulties of delivery, the efficiency, the off-target events. That's going to take a while, but it will be coming through, and I think we can be sure that's going to happen. And I think we can be relatively comfortable with the regulatory systems that are in place, which I think are listed on the next slide. As I said, reversibility would be a very nice addition to have even here. So if you make a mistake, if you could undo it, kill the cells, turn off whatever you've turned on, that would be very nice. And I know there are lots of things being worked on, and I guess you young guys are going to be the ones who solve that problem, how to do that. So I urge you to do that. But it raises the issue of enhancement, and I want to spend a bit more time on this. So we said in the report that some somatic genome editing should be used at the moment just for treatment or prevention of serious disease and disabilities, and not to make things, quote, better, to enhance things. But the question, this comes up here and it comes up again when we talk about germline editing, how do you make sure where you stop? How do you even define where you stop? What the difference between, if you've got a disease and you're trying to cure it, that's clear. If you've got a disease that exists and you're trying to prevent it, it's a little less clear. And where the borderline between prevention, let's say that therapy is cure and prevention. The borderline between prevention and enhancement is kind of blurry. A couple of examples up here. We have a lot of enhancements that we do right now. It's not just genome editing. This is not a specific problem with genome editing. It's a problem with a lot of modern technologies, robotics, neural interventions that are not genome editing, drugs. Comes up a lot in the context of athletics, which I think rather contaminates the question. That's a specific issue of a competition where you want an even playing field. But it comes up more profoundly in thinking about, is it going to be feasible for people to edit themselves or their children, somatically I'm talking about, to make themselves more efficient at something, to make themselves more beautiful, otherwise make themselves more successful. And this is a societal issue. It's not a technical issue. It's a question of how far should one go. And it's a very difficult problem because these boundaries are very difficult to define. But we have to think about it because it's with us now. And at the moment, the risk-benefit assessments, which is how all the regulatory agencies work, would preclude most enhancements, probably all enhancements. Because there are risks to this. We're still working them out. The off-target events seemed at the beginning of our discussion a year and a half ago to be serious. They're waning. And I'm sure you've heard about that during this week. It seems to be much less of an issue. And if you're doing, let's say, ex vivo editing, you're culturing cells, they're undergoing mutations as you culture them. There's a background rate of mutations. And it's getting to the point where the off-target events are getting down towards that rate. So one can't quite say that yet, and it's going to vary with different sites and different technologies. But if that's going to be true, and I think it will be true before too long, then the off-target events aren't going to be the problem. So in that case, the risks are going to drop. Now, if you're sick, the benefits are clear. If you're in an environment where there's a lot of infection by some disease and you want to prevent that infection, that's clear, and it's similar to vaccination. It's just done a different way. That seems okay. But let's say we're dealing with muscular dystrophy. You heard a discussion of that, which is a very helpful background for what I'm about to say. So muscular dystrophy is pretty complicated. It's a single gene, but the mutations are very variable. Many of them are spontaneous rather than inherited. So, and they don't only affect muscle. They affect heart and sometimes the brain as well, because dystrophin is expressed in a number of different tissues. So there are ways of ameliorating, in principle, there are ways of ameliorating muscular dystrophy by editing satellite stem cells for muscle so that they're bigger and stronger. Express myostatin, which is a TGF-beta family member, they get bigger. And you can make mice and cows. There's a mutant in cows that has more muscle, which is out there. It's in the territory that Hank was talking about. It's a natural mutant. It wasn't an engineered one. But you can engineer this, and you can do it in mice, and you get Schwarzenegger mice. You could do that for people, in principle. So you might get a thing approved by the FDA to ameliorate the symptoms of muscular dystrophy, not cure them, but ameliorate them. And that same construct could, in principle, be used for enhancement. So that brings up this off-label use question. So as you all, I'm sure, know, if a drug is approved by the FDA for one indication, doctors are allowed to prescribe it for others. And that's, in fact, important because a lot of advances happen because of that. They say, well, I should be able to ameliorate this disease, not the one for which it was approved. And I'll give you an example. Oh, it's gone. No, there it is. HGH, human growth hormone. That's a drug that was developed by, just down the road, by Genentech and used for people with growth hormone deficiency or other people who have way below normal stature. But once it started being used, then it started being used for people with sort of normal stature to make them taller. And then it was said that it inhibited aging and made people better. And it started to be used for all sorts of things in the standard off-label way. Some of those things were pretty unwise, and people had bad side effects. And in the end, Congress stepped in and regulated it because it needed to be regulated. So the same issue comes up here. Not with very many. It's not nearly as serious as it is for drugs because most CRISPR-Cas or other modern genetic engineering tools are designed for a specific process. The guide RNA is to make a particular change. And the CRISPR-Cas and delivery platforms are designed for a clinical use. And in most cases, except the one I gave you, the somatostatin one, the same tools cannot be used for enhancement. Then that's not what they're designed for, and they won't do it. So you'd have to get the FDA to approve it as a new entity, drug or mechanism. So that, at the moment, the risk-benefit ratio isn't going to be very good for that. The risks are the same as they are when you're trying to cure a disease, but the benefits are less evident. So for the time being, that's not going to be a huge problem. But as the risks drop, then this is going to become more of a problem. So we said, for now, no editing for enhancement. And that there needs to be a big public discussion about that, organized public discussion about that to find out where we want to draw the line. We already allow a lot of enhancements. Is there any reason why we shouldn't allow enhancements through genome editing? Perhaps not, but it's got to be discussed. In public opinion surveys, for what they're worth, there's a fairly strong consensus that people approve of somatic editing for treatment and prevention of disease, but they don't, on balance, approve of it for enhancement. It depends very much how the question's asked and what people understand. And it changes whether you ask, should it be done generally? Should scientists be allowed to do this? Or if you had to face this problem, what would you decide? People always approve of it rather more when it bears on their own interests. It's quite striking across all the surveys. And there's a table in the report that lists this. It was quite compelling. So this is a boundary that has yet to be drawn. And exactly how it's going to be drawn is not clear. And one of the things which we said repeatedly in this report about several things, this being the first, there needs to be broad public engagement into these questions. And I'll give you an example. When MRT, mitochondrial replacement therapy, was being debated in the UK, the government hired an outside entity to run a whole series of public engagement forums, workshops, feedback, public opinion surveys to find out what the public thought. And it went on for quite a while, and it played into the parliamentary decision. This was a legal regulatory decision by Parliament, taking into consideration what the public thought. And the public basically thought, well, if you can fix this, you should. And that was perhaps not expected, but it's a good example, I think one of the best examples of how public engagement should be done for some of these things. As Hank said, we need better regulations in several places, and we need public input into those. One of the things I thought about when you were talking about DIY, we didn't say much about DIY in the report. I looked into it at some length. There is a big community out there. It likes to think it's self-regulated. Obviously, it doesn't regulate everybody. There are rogue people out there, maybe. On the other hand, they are thinking about how to do it, and most of them are pretty responsible. I think they need to be engaged in this as part of the consideration about how this should be regulated. Same thing's true for enhancements in general. So that's a strong recommendation that we made both for this and, as you'll see, for germline editing. Public input is important.
Let's think this is just a bunch of crazies on the internet. There's a place about 40 minutes from here called BioCurious.
That's right.
Which is full of DIY computer scientists trying to learn PCR and growing E. coli and violating biosafety regulations by the dozens.
That's partly true, but it's also true that that community is trying to regulate itself. They're not doing it with any sanctions, right? I think that any of these things, to prohibit and contain anything, you need some sanctions. You need some penalty for breaking the rules, and they don't have much in the way that they can do. But I think they should be engaged. It's because there are some responsible people there, and there's some ignorant people there, and probably there's some irresponsible people there. And that's just picking up from Hank's example. I think that's an example of where there's a community out there that cares. In the case of somatic editing, there's a community of patient advocates who care a lot. And they know quite a lot. And their input is important. That's why we had such people on the committee, and we listened to a lot of them. It's helpful to know the different points of view. I think it's essential to know the different points of view. Okay, so I said that. So you probably all know this, but this is the key regulatory agency here is the FDA. You can't go to a clinical trial without their approval. And the same thing's true in Europe. And by and large, that works. But there are some things the FDA is going to have to learn, and there are some modifications that probably should be made. We didn't recommend them specifically, but there are some weaknesses in the way the FDA can control things. It's explicitly, the IRBs, for instance, are not allowed to consider societal impact. That's not part of their purview. They're explicitly told not to. So they're focused in on the individuals. The FDA has a bit more license to think about societal issues, but it's not going to drive anything. It's driven by risk-benefit. And I think it needs to be driven a bit more by input of societal impacts when we're talking about enhancement. So how that's going to be done is not clear. So using the existing regulatory mechanisms, they're pretty good. Limit trials to treatment and prevention of disease, no enhancement. That was the bottom line. Germline editing. This is the contentious one. As I said, we need to distinguish the non-heritable and the heritable, and I think we did that. So let's turn to the heritable germline editing. This is where you're editing germline cells with the goal of making an embryo, goal of making a child that doesn't have a genetically inherited disease. This can be done in animals. It's done all the time in mice. It's done in a lot of other animals as well. It's not yet technically feasible in human, but the techniques are advancing fast. And here I want to go into a bit of science to talk about how the advances are opening up possibilities that were not there a year ago, well, 2 years ago. But it raises, we'll come back to that, it raises a lot of societal issues as well as the technical ones. And even if the technical ones are resolved, 5, 10 years perhaps, something like that, I don't usually like putting a number on it, but in the not so distant future, then we're going to have to think about the non-technical issues, the societal ones, the ethical ones. And we should take advantage of that time and be thinking about this, not wait until we have to deal with it in an emergency way. And I think that that's really got to be worked up. This is back to public engagement and thinking about how to do this. So the question of off-target events, just briefly, I'll come back to this. When we started this, that seemed to be a block. That was what everybody was saying at the end of 2015. The off-target events are going to preclude this. Well, I think that's probably not true. They're dropping, the tools have been engineered so that the off-target rate has dropped a lot. There are ways of assessing it. Still need a bit of improvement, but there are ways of quantifying that and asking, have I done it cleanly enough or not? So this is not to be ignored, but it's likely not an insurmountable issue. So I'm going to pass over that. Happy to talk about it. Then it seemed mosaicism was going to kill this. If you edit an embryo, you heard about this in the case of editing cells for sickle cell anemia. Same question. Can you edit every cell in an embryo? So if you do the editing in a zygote or in a 2-cell stage, it is true in mice that you often get mosaics, so that, I colored these cells in, so that by the 8-cell stage, some of them are edited at one allele, and some of them are edited in both alleles, and some of them are not edited at all. So that happens. There's no question it happens. And in the Chinese experiments that triggered this alarm, that was what they saw. Lots of mosaicism. And it's true in mice, too. And there may be ways of improving that. There are some advances that suggest that it can be reduced. But there's no way of finding out whether you've really reduced it. If you edit here and you take a cell by PGD and ask, well, did I edit both alleles? If that's what I needed to do. That doesn't tell you that's true of all the cells, and you can't assay them all and still have an embryo to implant. So it kind of blocks it, and it seems unlikely that we'll get over that. But there are other ways of doing this, which have become much more realistic to think about in the last recent years. And this is editing the gametes, not the embryo. So this has become, particularly with spermatogonial stem cells, this has now been done quite extensively by a number of different groups, where you take spermatogonial stem cells, either take them out of the testis, or you generate them from HES or IPS cells, and then you can culture them in vitro, and you can edit them, you can validate them, you can grow them up and know they came from a founder cell that was properly edited, doesn't have lots of off-targets. You can then put that back in, either into the testes or in culture, and differentiate it. You get spermatids and sperm, and then by ICSI or IVF, you can fertilize an oocyte, make a zygote, and make an organism. This has been done in mice, rats, and monkeys. Hasn't yet been done in humans. People are thinking about it. It will probably come. There's little reason to think this wouldn't be feasible in humans. This is more recent and harder, because oocyte development is much more complicated, but you can do this too. You can take an adult cell, make an IPS cell, or you can take a blastocyst and make an HES cell. Let that develop to get primordial germ cells, which are pluripotent. And then there's a sequence of events here, which I won't go into. A lot of events that go on to make an oocyte progenitor, and then a mature fertilizable oocyte with polar bodies having got to meiosis II. And then you can take that oocyte and use it by ICSI or IVF for sperm, and you can get mice. And this works pretty efficiently. A couple of groups have done this, very nice papers, a couple of Japanese groups. And it hasn't been done yet in humans. It hasn't been done yet in very many places, but it certainly works in mice. So with less confidence than I said for spermatogonial stem cells, this seems a feasible prospect. People have been talking about this for a long time, and there have been a number of claims that are probably wrong, but this looks as if it may be feasible. We'll see. So if you edit here, you get mosaicism. But if you let that develop a bit further, you get primordial germ cells, or you make pluripotent cells of other kinds. Then you can go forward and make oocytes and make mice. Those are those two papers. Alternatively, you can let them go to the testis and make spermatogonial stem cells, edit and validate, like I said, expand and fertilize, and you're back there. And this has been used to correct a genetic disease. So there's a mouse line that has cataracts genetically. They went in and edited during this sequence the gene that caused the mutation that causes that back to normal and cured the cataracts in the mice. This is the paper. So this now seems to me, and I think to the committee in general, is the way this is likely to move forward. The spermatogonial stem cells, almost certainly. The oocytes, less certainly, but given the way things are moving fast in everything, wouldn't be unlikely. So technically, this is, I suspect, we think is going to happen. And as I said, it's time to think about what are we going to do about it. It's not going to happen this year, it's not going to happen next year, but it probably is going to happen over the next decade. So it's time to think about it and decide what should be done and what shouldn't be done. If it's feasible, is it approvable? So why might one want to do it? There are thousands of genetic diseases with simple mutations responsible for them. Individually, most of them are rare. Don mentioned several that are not quite so rare for one reason or another. Most of these are rare. There's probably no heterozygote advantage for most of them, like there is for the hemoglobinopathies, perhaps for CF. So they're less common. But when you think about the families that have to deal with this issue, we're talking about 5% of the population, the parents and the relatives and the siblings of children with a genetic disease. It's a major problem for those people. So what do? Well, they face difficult decisions, those people. Should they take the risk of passing on the inherited disease? Many people do. So the wish to have a genetically related child is strong, and in some societies is strongly endorsed by the culture, Israel being an example. And so people take the risk. Now, there are ways of mitigating that risk, which I'll come to in a minute. Alternative is to forego children or to adopt children. Perfectly reasonable option, but not for some people. They don't want to do that. What about various alternative reproductive therapies? PGD, pre-implantation genetic diagnosis. You make a lot of embryos by IVF. You go in there and do genetic testing of one blastomere and ask which of these embryos don't have the mutation, and implant those and throw the rest away. That's done for some things now, and it works. Some people don't approve of it because it involves throwing away embryos. Even earlier, prenatal genetic, and not earlier in the sequence, but earlier in time, prenatal genetic diagnosis is widely used for trisomy 21, where people look at the embryo and decide whether it's going to be a Down syndrome child or not. And some people are willing to abort the ones that are and preserve the others. So that's an alternative, right? At the moment, it's probably a better alternative. But some people disapprove. Some people don't want to do it that way. Or would germline editing, if it was feasible, which it probably will be, and safe, that might be an alternative option. And for some people, that might be the only option. The example that's usually used is Huntington's disease. It's a dominant genetic disease. One allele gives you the disease with penetrance close to 100%. There are other genes like that. There are a lot of collagen extracellular matrix genes which are dominant negative, and they're not that uncommon. And so you can't pick an embryo that avoids the genetic lesion. And then there are things like, I mentioned muscular dystrophy. If the mother has the gene, you're not going to cure the whole thing somatically. You could cure it by germline editing, because you get to all the tissues, and as Don mentioned, getting the delivery to all the muscle cells is not easy. That may get easier with appropriate targeting of the delivery system to get it into muscle. It probably won't go to the brain. It won't fix muscular dystrophy, because as I said, I think it's 1/3 of the muscular dystrophy patients are spontaneous mutations. So this is no help for those. So there are a large number of rare situations where this might be very useful. It's never going to be a large number for a foreseeable future, because it's expensive and there aren't very many that can't be dealt with by these alternative routes. But there are some. So that comes to the question, well, should you ban it for a few people for whom it's a good idea, or should you allow it? And that's a philosophical issue. It's a societal issue. I have an opinion. I'm sure each of you does as well, and it will change if it turns up in your family. So this is a complex issue. And this is, again, where we need public discussion. So what are the concerns? Obviously, the genetic changes, we always say maybe, because it depends how the genetics goes, but it probably will be inherited by the next generation. It's commonly viewed as unacceptable. It has been said for decades, perhaps most notably since Asilomar, where they said, you shouldn't do this. And everybody repeated it. They could say that because it was impossible. And so they did. It was a reasonable decision at that point. And it gets repeated over and over again, and it's embedded in legislation. But it's time to think about it. That was what we decided. We're not writing this report for the next 2 years. We're writing it for things going forward. What should we be thinking about? Whether or not that's a good idea. So the multi-generational risk could also be benefits. You could eliminate a rare disease from, let's say, an inbred population. Huntington's disease would be a good example, where it's quite frequent in small populations. And there are some others like that. If you're going to do that, you need long-term follow-up on the earliest ones to find out how things are, whether you've made a mistake. That's difficult, but it's not impossible, actually. It's been done for some other interventions to have long-term multi-generational follow-up. The question gets raised about there's no consent by the affected individuals. That's also been raised in other contexts, and it isn't really a problem. I mean, people make decisions for their children all the time, to which the children don't consent. And Hank may be able to help me out here. It's been made clear in other situations that that's not a serious issue, although people worry about it. More diffused as a worry is the lack of intervention, the risks of intervention in nature. Now, obviously, there are technical risks, which one needs to deal with. But this is a more philosophical one. We shouldn't be doing this. It's playing God. That sort of position. Now, that's very hard to rebut. It's a often religious. Otherwise, it's a cultural, societal objection, which can't be ignored. It's the same thing as with stem cells. People didn't agree with making human stem cells for philosophical reasons. That didn't mean, and it was decided that it didn't mean, that they could ban everybody else from doing it. Is that true here too? It's a similar sort of argument, but it needs to be discussed. People have raised the question about whether the ability to edit out genetic mutations that lead to children with severe disease will lead to the pressure on people to do it, that it will be negligent not to do it. I mean, That's a valid concern. In fact, when you look at what's happened with IVF and other ARTs, it hasn't happened that people are pressured to do things. People worry that it's going to affect the acceptance of people with disabilities. That hasn't happened either. There are some communities, for example, the deaf community, a lot of genetic diseases, single gene diseases that cause deafness, quite a number of them, but well understood. But that community, many of the people in that community don't want to be considered disabled and would not want to edit out that gene. And obviously, one shouldn't make them. But one needs to make sure the pressure from insurance or something like that doesn't lead to that. And these issues are hard to handle. They're diffuse, but they're important. This one is the one that comes up a lot. That you see this in the headlines, designer babies. This is going to open the way to making people who are more intelligent, and the rich will be able to do it and the poor won't. So my response to that right now is we know lots of ways of making people stupid, but we don't know very many ways of making them smarter by genetic engineering, but that will change. This is part of the slippery slope argument that once you start doing something for probably agreed good reasons, then you open the technology towards doing them for less good reasons. And I don't think one can dismiss that issue. I think what one has to think about is how to slow that down. And I don't believe you can stop it completely because of the international aspect, which I'll come to in a minute. How are we doing? Yeah, a little bit of time. This one, the inequality of access, that some people will be able to afford to do this. And because it's genetic, that group of people will then sort of have embedded that advantage in that group and made a permanent change in the distribution of power, influence, money, whatever. I don't think that can be ignored, but I don't think it can be solved by us. That's a societal problem. I mean, we had a discussion a little bit about this over dinner last night. I mean, if there was a single-payer, government-supported health industry, this would become less of a problem. It would be good for many reasons, this being one of them. So, and it isn't as much of a problem and isn't perceived as much of a problem in Europe because they all have socialized medicine. So that's one of the examples of the different categories. But again, it's something that needs to be discussed. So, okay. So I was going to say that that's, again, a place where we need to engage public involvement. So one of the things we said was that, as was done for the Human Genome Program, funding that goes for science of human genome editing should include, as part of the same programs, maybe not the same grants, but the same programs, some scientific, well, evidence-based investigation of what the public thinks about this. Not just to educate them. The idea that it's a matter of educating the public who are ignorant, that's not the whole story. It's finding out what people think. It is partly that, but it's also importantly hearing from the public what they think and incorporating that into how you do the rate. So as I gave the example of MRT in the UK, there are some other examples which we discuss in the report where Denmark has done this fairly effectively over a long time. France tried it and did it pretty ineffectively for a specific thing. So there are things to be learned from the other countries as to how they've done this. And it needs to be done. This shouldn't be a decision made by regulators and scientists without input from everybody else. So right now, you can't do clinical trials on germline editing in the US, not because of any law, but because of the funding of the FDA. So there was a rider put in on the FDA budget 2 cycles ago in the continuation resolution that said the FDA cannot consider, can't even answer and acknowledge requests to do germline editing. And this was stimulated by the mitochondrial replacement therapy. So it blocked that completely. It was renewed in April when that continuing resolution was renewed. So there's a historical precedent here about this with respect to embryo research. As you may know about the Dickey-Wicker Amendment, which is a rider that's been put on the HHS funding for many years. It's in there, and it goes in there every year. It says basically says you can't do anything to an embryo that's not for its own good with federal funding. So it's blocked federal funding for embryo research for a long time. Once these things are in there, they're hard to get out because that has to go through Congress to take it out. And there's a debate that's, as somebody said earlier today, it's not going to be possible to get Congress to undo that. So there's a good chance this will continue for a while. So that this actually wouldn't be allowed anyway. It's blocking MRT at the moment. Most other countries currently prohibit germline editing. And as I said, some of this is historical. And I don't think it's inappropriate right now. But the question is, how long should that stay in place? And what does it take? So we made a list of things that would need to be done to make it feasible to think about approving this. Much more research, strict oversight, and a specific set of criteria that would need to be met. And this is 10 criteria that need to be met. Absence of reasonable, acceptable alternatives. That was the question about, can you do this by PGD? If you can, maybe that's a better way to do it, etc. Edit only genes that have caused serious disease or disability, and that you understand, and that you've convincingly shown that they cause or strongly contributory. So not just any gene, not going in and editing something like ApoE4, which does contribute to Alzheimer's. Exactly how, we don't know. We don't understand why that's true. That's not a candidate. But something like Huntington's disease would be, just to put two extremes there. To convert any, if you're going to do this, you have to do it by converting it to a gene variant that's widely distributed in the population and not known to cause any disease. Not just to anything, not just to something you think might improve it, but if you like, back to normal. There's a lot of discussion in the report about what normal means, because that's not well defined either. So lots of, obviously, preclinical and clinical data. Rigorous oversight. So this is going to have to be done by somebody, not quite clear who. Probably the FDA, but the FDA would need more expertise than it has, and it would need to incorporate societal input. Comprehensive plans for the multi-generational follow-up, and a continuing reassessment with public participation. And reliable oversight mechanisms to stop the slippery slope without serious debate. Now, what we said about this is, if these 10 criteria cannot be met, it shouldn't happen. And some people think those 10 criteria can never be met, and that's a debate we're going to have. There are a lot of people who think you can't possibly deal with a slippery slope, for example. And I think that's open to debate, but now's the time to think about it. How are we going to do this? It's related to what Hank said about how do you regulate some of these other activities using CRISPR. Can you or can you not stop them if you think they're a bad idea? So the key messages of the report as a whole: basic research in somatic gene therapy continue as they are. The existence of CRISPR-Cas and zinc fingers and talons doesn't change that at the moment. It's adequately regulated, but it should only be used for treatment and prevention of disease and not for enhancement until we've discussed this at length and come to some societal. Consensus is a difficult word. Some societal set of opinions that we can act upon. We're not going to get consensus, which if you look it up means unanimity, although that's not how it's usually used. That is what it means. And there isn't going to be that unanimity about this. But it's worked for other things like IVF, well, that wasn't really regulated, like stem cells and embryo research. It's going to be similar here. I've already gone through this, I think. So the bottom line is, we think this should be used to make healthy children, not designer babies. And we need a lot of international coordination to make this work, because while MRT, I said, is blocked here in the US right now by congressional action, people from here go to other places and do it. And there's a lot of stem cell tourism around the world where things that can't be done here, haven't been approved here, are being done elsewhere. That's going to be very difficult to block completely. And as I said earlier, there's not a good prospect that we could have a UN resolution that blocks it. And different countries are going to already do have different regulations, and they're going to, going forward, have different rules about what's done. How are we going to get those to be coordinated? So I'm not going to take a lot of time to go through this. There's a lot more underneath these bullet points. But we set out a set of fairly obvious criteria that should, general principles of how this should be done. I was looking recently at the International Stem Cell Society report. They have a similar list, very similar set of criteria. And I think that those, while generally agreed among scientists and clinicians, need to percolate into any regulations. They shouldn't be inconsistent with this. They can vary at the lower level. And these are spelled out in more detail. What does this actually mean? They're spelled out in more detail. So any nation that's considering allowing this should think about all these things and incorporate them into their regulations. So I'm going to stop now so you can ask some questions. These are the sponsors, including, you note, DARPA, which is an interesting question. I want to thank Altef, with whom I spent many, many hours over the past year and a half, and the other committee members, and many speakers and participants that we invited to the various meetings here and in Europe. The reviewers, a couple of whom are here, of the report, who were very helpful in enhancing the final product. Very, very good, helpful reviews. And the Academy leadership. So that came out, the final version just came out today or yesterday. So I'm happy to answer questions. So. Somebody must have some opinions or disagreements. So.
There's a student in the back here with a question.
Hank.
So this wasn't within what the committee was supposed to report on, but do you have personal views that you'd like to share about the political viability of the committee recommendations?
Well, that's a prediction, which I don't know. It'll depend on which administration. Going back to the stem cell one, which similarly pushed the boundaries beyond what was allowed at the time. It was during the Bush administration, who was adamantly against making human embryonic stem cells. That set of recommendations was taken over by CRM in California and turned into law. When Obama came in, it was turned into the regulations at NIH and was the basis for the ISSCR recommendations. So I think over time, my hope certainly is that this will make its way into more formal regulations. I'm surprised how little opposition there's been, actually, and I think that may be because of the political noise background at the moment. Other people are more worried about other things. I imagine it will come up again. The people who you would expect to object to it did. The people who don't approve of any of these things, ART, genetic engineering of any kind, don't like this. I didn't expect them to. Otherwise, it's been fairly well accepted by the scientists, at least, including the ones who don't really agree with it. They think it's a reasonable balance. So I'm hopeful. Can I predict what's going to happen? No. But my feeling is that there was no point in our saying things that only would extend for a year or two. We have to look out into the future and think about how we're going to deal with these things when they come along, which they will. This is going to happen, and just banning it won't work. And so we need to think about how to regulate it and set a good example. The countries that regulate things well, UK, US, do set examples for other countries as to how to do it. Professional societies and clinical groups and their policies are affected by this sort of thing, and they're probably one of the best ways of imposing discipline, which is why I brought up the DIY people. The people who are actually doing it often think fairly carefully, many of them, about how to regulate things. And that's been the case with stem cell research, even with IVF, which was totally unregulated when it came in. It still is, really, formally. So I think it'll influence those things.
There's another question over
Yep.
here to get to next.
Yeah, so you mentioned like engagement of public opinion. Can you explain a little bit more about what's the form of public engagement and how is this going to be perceived?
So there's a whole chapter on that in the report, looking at the ways in which it's been done. And I don't think that provides the roadmap for exactly how. It tells you some things that have worked and some things that didn't work very well. As I said, it needs to be two-way. It's not just the scientists and the clinicians educating the public. That's part of it, but it's nowhere near enough. And the ways that have worked best have been panels where significant numbers of people were involved and engaged in discussing, being educated and discussing, and then publicizing the results, doing formal public opinion surveys properly, and finding out what people really think. Some of those have been done, but that needs to be more organized. And having input in, I mean, there is already input into the RAC, for example, the Recombinant Advisory Committee. Public input, it's solicited. It could be a lot better. There should be more engagement, more opportunity for the public, and not just the vested interests, pro or con, who tend to dominate these things. That has to be, they have to be entertained, they have to be included. But we need to broaden the pool from whom one is collecting feedback. And one needs to pay attention to it. One of the things that was wrong with the French example is they did all of that to one degree or another, and then they ignored it. When they wrote the regulations, they basically ignored what people had said, and that caused a lot of, disapproval, reasonably enough. So.
Jennifer?
So at the beginning of his talk said he agreed with most of the report. I'm curious to hear what part he doesn't agree with and what your respective opinions are.
Yeah.
Just very little. I would have been a little bit more aggressive on pushing against the argument that, pushing the idea that germline modification goes on all the time, naturally.
So let me answer. I would have too, but this was a consensus committee. So.
And the other thing is, I think the limits on disease modification, germline modifications with respect to disease, to those for which that's essential, as opposed, what exactly that'll turn out to mean is unclear to me. If it's proven sufficiently safe, I don't know that, which gets into a whole other question about what sufficiently safe is. I'm not sure I would require people who could go through PGD to go through PGD rather than going through genome editing.
We didn't require it.
Well, the language is.
The language was very carefully crafted not to require it, but not to, to leave it up to people to decide for themselves. But we had to balance it. So I agree with you. I think that in a democracy like this, people should be allowed to do what's reasonable in their own context for their own reasons. You shouldn't ban people from doing something because you disapprove of it, if it doesn't cause damage beyond that. But that's where the societal issues come up in this. The germline editing isn't just the participants. It has the potential to propagate. And that means that there's more reason to incorporate the concerns of X about what Y is doing than in the case of somatic genome editing, where it's a risk-benefit question. Then people have to decide what they want to do for themselves, and they don't have to worry about the rest of the world. So that was why that was very carefully worded after long debate about exactly what we said. So in those 10 criteria. I'm personally more...
Carefully enough worded that I I actually read it differently from the way you apparently intended it.
Well, this was a committee of many people, right?
You plural, second person.
Yeah. Point taken. Yeah.
Would you mind if I used one of your slides to reinforce some of your points?
Sure.
Number 15.
15. Okay. What's that?
This one here, I refer to this as attribution. To whom do we attribute the need to be fixed? And there's something I learned at that international conference in 2015. I hadn't thought about it before, but the example you used of hereditary deafness, I often use. The one I used here on Monday was hereditary short stature. And you know, high-functioning people have you know, sort of non-standard phenotypes, but they don't need to be fixed necessarily. And so we have to be very careful about to whom we attribute the need to be fixed. This inequality of access I call distribution, and used the sickle cell example, where we may have a very expensive therapy before long that we can apply to sickle cell sufferers in Oakland, but the huge majority of people who suffer from sickle cell disease live in places where an expensive therapy is not going to be effective. And so, again, a societal issue, we need to be thinking about ways, both the scientists need to be thinking about ways that to make these therapies cheaper and more easily distributed. And the final one I wanted to reinforce was this issue, and I think it's true of all of the genetically modified organisms that are going to go out into the world. It's true of the plants and the animals, livestock, and humans, that you can't do adequate preliminary experiments to completely confidently predict the outcome in terms of phenotype. And in food organisms, no company is going to put a food organism on the market that has accidentally internally generated a poison. I mean, that's nuts. But there are going to be phenotypic consequences that are unpredictable in model experiments that we will only see in the real application. And these human medical applications is one area where that's
Yes, it is.
very true.
The MRT report said the same thing, that long-term follow-up is going to be necessary.
Yep.
And it has been done in other situations. You obviously can't force somebody to continue to participate over a long period of time. But it turns out that a significant proportion of people do. They want to participate in the long-term follow-up. It's not going to be easy, but it's going to be necessary. So, and on the inequality of access, I completely agree. These are all expensive technologies. They are going to be exploited in well-financed places like here, or by people with lots of money. Initially, that's always the case. It's in the Bible. Unto him that hath shall be given, right? And from him that hath not will be taken even that they have. So it's been around for a long time, and we're not going to change that. But that was why I said what I said about socialized medicine. I think that that does mitigate some of this problem, and I think some other problems as well. And that's a societal issue. It's not one we can solve in this community, but we shouldn't ignore it. So.
I'm curious, your last 2 points here on designer babies and slippery slopes. They're sort of related points. Do you worry? Are there two?
They are related. They are related points.
One that's like ethical and regulated within society, and one that's like as technical advances move closer and closer, you could enable sort of unregulated black market.
Well, no, they're those two things are different. The slippery slope argument is really, once you've approved doing one thing, it makes it easier to prove something that you wouldn't earlier have thought about. I mean, lots of examples. IVF was introduced to deal with fallopian tube blockage. It wasn't introduced to make babies when you couldn't have them yourself. It rapidly expanded into other things. And that's true of most advances, actually. once people realize you can do it, and we heard some examples of that before about how you could do that in agriculture and animal. So those are things you can't necessarily anticipate, but they're going to make more likely if one starts down the road for certainly what are good reasons. I could give you a lot of examples of that, but I won't take the time. The other one about rogue participants, which Hank also brought up, that's a different one. If you've got a regulatory system that draws a line and then the line moves, that's the slippery slope. The other one is if you've got a technology, which we have, that people could abuse, that's with us right now. And it's much harder to deal with. And the stem cell tourism and genome editing tourism that might happen is a separate problem, I think. So, I mean, it's a different problem. So.
Just as a follow-up question, I'm curious, did the committee, what was the canonical example of recent society that people use as a slippery slope? Would it be the approval of abortion through Roe v. Wade and then
It depends. It depends who you listen to. I mean, these concerns get talked about a lot. They've been talked about continuously in the ethical community. Hank would be better positioned to answer some of these questions than I am. The press seizes on them. Terms like designer babies, which go way beyond anything that's likely to happen in the near future. Not totally unrealistic down the road, decades down the road, but really not worth worrying about right now. But that's at the bottom of the slippery slope, or far down the slippery slope. In my view, a lot of these things are fears that don't get realized. In many of these prior cases where people have brought up things that would happen, I briefly mentioned a couple of them, and they are discussed in the report. People said if there was PGD, people would be using it a lot for the wrong sort of thing, and it happens. People use it for sex selection. It's not a good idea. So that's an example where it's true. But the idea that people would use it to select for lots of other things, or that they'll use this to make people with blue eyes, or keep their hair, or one thing or another, they're not totally ridiculous, but are people going to spend much money to get a kid with blue eyes? I doubt it. No. If it was the National Health Service, would they pay for that? No. So.
Fascinating discussion.
Yeah, it's
Summary
a long discussion, which we're going to have for the next decade. So.
Thank you again.
Okay.